a heme group in here. We'll get to the structure of heme a little later. And those red dots are iron atoms. Here's one that's mostly beta, but some alpha helix here. This is, let's see, UDP N-acetylglucosamine acyl transferase. It's involved in collagen biosynthesis. And so again, we basically hear a whole bunch. Here's a whole bunch of parallel beta sheet in here. See a whole bunch of parallel beta sheet in here, and then there's a couple alpha helices in a loop that's around. It's an enzyme. Catalyst. Here's one. This is pretty cool. Concatenol A. This is a lectin. Actually, you'll see these for 162. You do work with a little bit of lectin. So this protein binds carbohydrate residues very tightly, and they play a role in recognition and defense. Well, we're going to talk about this protein in just a little bit. Peptidylprolyl cis-trans isomerase. This again is it's a catalyst. It's an enzyme, and what it does is it binds other proteins where there's a proline and make sure that the proline is the proper cis-trans configuration. We're going to go over that in detail here in a little bit. Again, a lot of beta, pretty much all beta sheet in here. Here's some more, mostly beta. Oh, cal gamma crystalline. So this is the protein in the lens of the eye that forms the lens and allows refraction of the light. And there, this is from cal. And again, what happens with cataracts is this protein begins to lose its structure and misfold, adopt an invariant structure, and it no longer transmits light as well. Here's green fluorescent. Here's an example of a beta barrel, where the beta sheet flips around itself down in here. And in the middle, there's 3 amino acids that actually are post-translationally modified to allow them to fluoresce. And so this green fluorescent protein is important for jellyfish, but from a technological standpoint, it's used as a marker a lot. Yeah.
I'm guessing that those amino acids that you're referring to. Wait, did you say amino acids or protein?
Yeah, well, 3 amino acid residues within the protein. I can't remember what they are off the top of my head.
Oh.
But they end up, and I think the aromatic ones are involved, but I can't remember the structure off the top of my head. Retinol binding protein. So this also involves some sort of lipid transit through the bloodstream. Retinol is an important dietary component, an important basically vitamin in here, but it's a very hydrophobic molecule, so it has to be transported through the bloodstream. This protein binds it. You see it right here, binds it in there. So again, we have mostly beta sheet here, and then just a little bit of alpha helix. Flavin mononucleotide oxidoreductase. This is involved in energy metabolism. This is sort of a classic. This would be nice alpha beta protein, classic enzyme in here. They don't even say which enzyme this is. Tryptophan biosynthesis enzyme. I'm not sure, but we see the alpha helix here. See how we have the alpha helix here, and then the beta sheet in the middle. What that does is creates a cleft basically inside where you can have then a specific structure that allows for the catalysis to occur. We're going to get into that in quite a bit of detail here. Adenylyl kinase, this adds a phosphate to ADP to make ATP. Again, this is nice alpha beta protein. Flavoredoxin, this is an electron carrier here in energy metabolism. Alpha beta protein. And we kind of keep going. Thioredoxin, this reduces disulfide bonds. Again, alpha beta protein here. I'll just do a couple more of these here. Arabinose binding protein that binds the sugar here. This actually kind of shows, this is where the sugar is, the arabinose is here. You see how the structure basically forms a cleft to do that. So again, by kind of cruising through here, thiodisulfide oxidoreductase, this is another one involved in protein structural rearrangement. Just by going through here, again, we kind of remind ourselves you know, there's, in any given cell, human cell, there's probably 10,000 different proteins. So we can scroll through and we can basically see how all 10,000 of these proteins, they have different primary sequences that ultimately result in them folding up in a particular manner to enable them to do what they're doing. Oh, this is pillin. So what happens with this protein here is this alpha helix down here is a hydrophobic alpha helix, and it'll insert itself into a membrane there. Triose phosphate isomerase, this is a glycolytic enzyme. And again, what we see alpha helix kind of surrounding a beta barrel type structure in here, and then there's a cleft in the middle in which the enzyme catalysis. Anyway, we'll stop there, but just to kind of emphasize that point. All right, so can't remember exactly where I let off last time, but we're basically talking about tertiary structure. Again, overall spatial arrangement. Of a single polypeptide. If we talk about major structural classes, then we have alpha, mostly alpha helix, beta, mostly beta sheet, and alpha beta, which is a mixture. And again, structural biochemists have tried to go through and make further subdivisions in there to try to provide additional hierarchy, but it's a pretty rough, loose hierarchy. Now, one of the other things that I wanted to mention too was structural domains. And this is, sort of the examples of what I've shown you here is you have a single polypeptide and it adopts its appropriate secondary structure, and then it kind of comes in it, and then it forms its overall tertiary structure. Now, sometimes, actually many times, there are different parts of the protein will basically fold independently. We'll end up adopting structure, and so we call these different parts structural domains. And it really comes from the evolution of proteins, protein evolution. That in many cases, many proteins have actually evolutionarily arrived from the fusion of two other proteins. So there might be two proteins doing something, and those proteins end up getting fused. So there's a single gene that encodes a larger protein that used to be two separate proteins. And when we get to metabolism, we'll talk about that, why that is. But basically provides essentially if you have, say, two enzymes that are closely related in a biosynthetic pathway, it basically puts those two enzymes on the same polypeptide, and they can move forward. Or if you have one protein that's say important for binding ATP, and another protein that's important as a catalysis, you can kind of link those two functions together. Anyway, so what happens is when those get fused, the two different parts of the protein will fold independently. One will fold like its sort of ancestral precursor, and the other will fold like its ancestral precursor. And so while you have a single polypeptide, within that single polypeptide, you sort of have two separate functional folding units, okay? And those functional folding units are known as domains. So these are independently folding regions on a single polypeptide. Example is the human fatty acid synthase. This is an enzyme that builds fatty acids. There's actually about 8 or 9 separate catalytic activities that that are required to build a fatty acid, and each of those catalytic activities actually has its own structural domain, but it's all part of a single polypeptide. All right. We should kind of also review what are the main forces that are actually driving the tertiary structure. What are the stabilizing forces? You know, why do those bunch of alpha helices fold up in one particular way versus another, and what's actually holding them to adopt that particular structure? And probably the biggest is the hydrophobic effect. That when a protein overall folds, you basically the hydrophobic residue side chains get buried on the inside, right, away from the solvent. We talked about what drove the hydrophobic effect. It's an entropically driven process. Okay, so that's big. But then there are other things. There are charge-charge interactions. These are, for example, salt bridges. An example might be if you have an aspartyl residue here, might interact with a lysyl residue. So that kind of stabilizes it once it's there. To a lesser extent, our internal hydrogen bonds and also van der Waals interactions. You know, we talked about how these are pretty weak interactions anyway. But on the other hand, you got a lot of them. When a protein folds up, there's got a lot of them, and so they add up. Now finally, we can get disulfide bonds. We talked about disulfide bonds here. Disulfide bonds are probably more important for once a protein has adopted a structure, for locking it in place. So if we look at disulfide bonds, here's our only covalent, of all these different interactions, is the only covalent bond. But these are important for locking locking the structure in place. And it's usually, mostly associated with excreted proteins. So insulin is excreted out of the beta cells into the bloodstream. So it has some disulfide bonds holding its structure into place. There's a couple reasons for that. One is that within the cell, the overall environment is pretty reducing, and so the disulfide bonds would tend to get reduced rather than stay oxidized. And then also they don't need it. Sort of like, okay, if a protein is going out into the harsh oxidizing environment outside the cell, then it needs some help locking its structure into place, and so it will form a disulfide bond. But if it's hanging out mostly inside in the relatively mellow environment in the cytoplasm, it doesn't necessarily need it. I'm going to go and show you something that your book doesn't really get into, but it's kind of cool. It's like, I showed you all those structures. Well, how did people get those structures? And show you the process. The major way of doing it is X-ray crystallography. And it's a pretty cool process. You need a lot of very pure protein to do that. So, the very original X-ray crystal structures were obtained from things like, hemoglobin and that sort of stuff, where you had an easy source of a lot of protein, and the protein was easily purified from its native source. These days, mostly what you do is these heterologous protein expression systems, where you take the gene, clone it into E. coli. We talked about this before. Make E. coli, have E. coli, make a whole bunch of it, and then purify the protein, and then use that to crystallize the structure. So how does this actually work? What you do is you take a drop of your purified protein and hang it from a slide here, okay? And then you have this reservoir solution that actually has a higher ionic strength. And then what happens is the water slowly evaporates away from the drop and then comes down and it enters the reservoir solution. So your concentration of protein very, very slowly gets higher and higher on this drop. Okay. Now the whole trick is there's a whole bunch of different recipes for basically what else is in your with your protein, what other sort of buffer solutions and that sort of thing are within your protein, and what's in your reservoir solution. And that's some of the trick. I mean, there'll be like people who do this might have 100 or 200 different recipes for different crystallization media, and so you'll take your protein, you'll set up one of these drops, and a bunch using 96 well plates, and basically try a whole bunch of different ones until you find the one that works for your particular protein. Yeah, so here it's actually this is what a 24 well plate here, but you basically set them up in here. And then, you basically, here's on your shelf, you've got a whole bunch of, different conditions and that sort of stuff, and you're hoping that your protein crystallizes. It's kind of a numbers game, to tell you the truth. Some proteins crystallize really easy, some of them don't. Then what you could do is after a while, like weeks, maybe several weeks, you start looking through the microscope to see if you have any crystals being made. And then if you're lucky, you'll get things that look like this, rectangles, daggers, that sort of thing. But you don't want these really needly crystals. Once you get those crystals, you have to harvest them, okay? And then you have this loop, and you store the crystals in liquid nitrogen. And then usually once you harvest the crystals, you use X-ray. So the basic idea is you have X-rays, you go through the crystal, you get this crystallization pattern. then you go through some fancy math basically here, you end up with an electron density map, and then from the electron density map, then you try to model a generated structural model here. Where do you get the X-rays? You can get, in-house you can get low resolution X-rays, and so just to kind of see if your proteins are any good, or your crystals are any good, you might do it in-house, and just to see it, but that doesn't really give you, that'll tell you, okay, are your crystals really diffracting? But you really want some high resolution, you need like a really powerful X-ray source. So what you do is you go someplace like down to the Stanford National Accelerator, where they basically, they have an accelerator that comes down in a loop, and they get these, now I can't even remember what they accelerate, I'm not a physicist here. But basically, they get this thing going, all this was designed to do higher energy physics experiments, but what they found when they were doing the high energy physics experiments is, boy, they generated a ton of X-rays when they were doing this experiment. And so, doing these experiments, so somebody said, hey, I know I need some X-rays, so let's harvest those X-rays so I can do crystallographic studies. And so you basically go into this linear accelerator, and they basically have this area down there in which we basically harvested the X-rays from these linear accelerator experiments to be able to use for crystallography. Yeah, geek heaven. And then you basically sit at a computer there, and there's your loop down there in the bottom left. You put the crystal in, and then you hope the heck that it all works. And then if you're lucky, you start getting a good diffraction pattern here. And when you get that good diffraction pattern, then you get, when you get that nice diffraction pattern, then there's the technique of actually turning that diffraction pattern actually into a structure. And that's basically greatly facilitated by various different computer programs, and I won't say you just click and click and click necessarily, but Dave Wilson, the crystallographer over at UC Davis, who we've worked with several times, he takes undergraduates down there, and they can do it. So it's not something that's inaccessible. And then you end up getting you get the structure. Here's an example why I have this of the domain. So this protein might be a single polypeptide, but what you really see is that it's kind of four separate, independently folding domains, all within a chain. What else? Okay, we'll hold that for later.
So I have a question. So those domains, those be the secondary proteins that make up the tertiary, or?
No, you would really consider that. I mean, it's where the hierarchy breaks down a little bit, but you basically, I think that a domain is different than a super secondary. We talked about motifs, where they're basically just common ways a couple of secondary structures fold. A domain is really an independently folding part of the tertiary structure. Yeah.
I remember back when reading double helix, one of the problems Watson had was his mass background wasn't as strong.
Yeah.
So, and I also remember like reading one of the papers, they were talking about like a Bessel function, which I don't know. So like, I think my question is like, how bad was it back then, like compared to now? If you say with programs.
Oh, in order to be able to, like this, that's a good question. I don't know how, how tough it was before all the computer algorithms actually do the deconvolution. Yeah, that's a good question before the crystallography. All right. X-ray crystallography isn't the only method. You can also do their NMR. It tends to be lower resolution, or you get relatively small peptides. There are some different things. Okay. Now I want to get in and talk about quaternary structure, the final element of structure. most proteins greater than 100 kilodaltons are actually formed of multiple peptides. Basically, two different gene products end up coming together and associated to make the final functional protein. And there are a lot of proteins that are greater than 100 kilodaltons. So there are a lot of proteins that are like this. So example, EcoR1, the restriction enzyme. We talked about restriction enzymes, right? These are the enzymes that cleave DNA in a sequence-specific manner. It works as a homodimer, which means it's a dimer. Di means that there's two subunits. They're two independent polypeptides. And homo means that they're actually the same. So homo, the same. Dimer means two polypeptides. And that would be signified by saying it has an alpha 2 structure, meaning alpha refers to the single subunit and that there are two of them. Another example is hemoglobin. Protein that binds heme, which carries oxygen through the blood and carbon dioxide through the blood. Okay, it is a heterotetramer. That means there are different subunits. And that there are four polypeptides. Now it turns out all four of the subunits aren't the same. They basically have two of one and two of the other. So it has an alpha 2, beta 2 structure.
And the alpha and the beta is just, will be more specific with the subunits?
Yeah, so the alpha and beta are associated with the different polypeptides. So this is one polypeptide, this is another polypeptide. The one thing it's not, okay, is because it doesn't have anything to do with alpha helix and beta sheet. So there's a potential for confusion there. But it just says that there are two of one subunit and two of another. If you had a third subunit, you would call it a gamma subunit. You know, if you had a third polypeptide. Another example is, he's going to call it Rubisco. This is the world's most abundant enzyme. It's the enzyme that's responsible for fixing carbon during photosynthesis. Okay, and it has an alpha 8, beta 8 structure. So there are 8 of one type of subunit and 8 of another type of subunit. This subunit is bigger than this one, and so it's also known as the large subunit, the small subunit. But there are 8 copies of each in the final functional protein. Now the interesting thing about this is, you know, again, these are separate polypeptides. The interesting thing about Rubisco is the large subunit, the alpha subunit, is actually encoded by the chloroplast. Remember, the chloroplast is an organelle, has its own DNA, so it's actually encoded in the chloroplast, and then it's translated within the chloroplast by the chloroplast ribosome. whereas the small subunit is encoded in the nucleus, and then it's translated, it's made in the ribosomes out in the cytosol, and then that small subunit actually has to be transported into the chloroplast, where it mixes with the large subunit to make the final functional protein. So this is actually kind of common. A lot of mitochondrial proteins are the same way, that their parts of the proteins are actually encoded by the mitochondrial DNA and made within the mitochondria, and parts of the protein are actually encoded by the nucleus and then get imported into the mitochondria. Nothing's so simple. So then, just as another example to kind of show you a little bit more extreme, here is one of the most abundant proteins around. It's called complex 1 of the electron transport chain. Sits in on the intermitochondrial membrane. We're going to talk about it in second semester. Transport. There are 45 different subunits. It's a very complex structure. 36 are nuclear encoded. Nuclear encoded, translated out in the cytoplasm, in the ribosomes out in the cytoplasm, then imported into the mitochondria, and then 9 of them are mitochondrial encoded. And this is basically I right at the beginning, right, I showed you all these pictures of these relatively simple single polypeptide proteins. So you kind of get that model in your head. Then you got to step back and remember that's sort of the simple end of the spectrum, that a lot of proteins are much more complex and involve a whole bunch of different polypeptides that all come together to make the functional protein. Okay, the only other point I want to make about the quaternary structure is that most of the interactions is really that allow these associations of these. Most of those are held together by non-covalent forces. The only covalent force that we saw could play a role in tertiary structure was the disulfide bond. And disulfide bonds typically don't play a role in quaternary structure of proteins. All right, I wanted to get through that. Your textbook kind of takes a sidetrack a little bit. It doesn't quite go through that linear order of primary, secondary, tertiary, quaternary structure. I kind of wanted to present it as boom, 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 boom. There's right in the middle there, your text sort of takes a sidetrack and then starts talking about the structure of fibrous proteins. Because so far, really what we've talked about here are the structure of what we call globular proteins. These are the sorts of things that I've been showing pictures of. But there's a whole other sort of set of proteins that basically mostly sort of have secondary structure that are fibrous proteins. This includes keratin, the protein that makes up our hair and our fingernails. It includes collagen that makes up our cartilage, and as well as things like silk fibroin that actually make up spider silk, and then we use as a textile. All right, so now I'm going to kind of go back a little bit, and I'm going to talk about the structure of fibrous proteins. first we'll talk about keratin, alpha keratin. Again, this is hair. Fingernails. You want to get out of the human domain. You know, whales, they eat the baleen whales that are filter feeders, things like the gray whale and the blue whale and these sorts of things. They when they go and they scoop up large amounts of either small fish or amphipods, shrimp-like kind of things, and they basically filter them out. And the baleen that hangs from their mouth that acts as a filter is made up of keratin. So you know, just to kind of remind you, it's not all about us, mostly about us. Basically, what we have here are two alpha helices. I'm going to show you some pictures. Form a left-handed coiled coil dimer. You have two alpha helices that, you know, the alpha helices are right-handed, but they twist around each other to the left and form this dimer. And then you basically, it's kind of like making a fabric or something like that. Is you take these coiled coils and you weave a couple of those, and then next is two coiled coils. Form a protofibril. And then four protofibrils form a microfibril. We're basically spinning yarn here. And the microfibrils associate form a macrofibril. And then all these macrofibrils are stabilized by disulfides. All right. So again, you know, here's our alpha helix. We get two of these alpha helix that twist around in a left-handed way to form the coiled coil. The coiled coils form these protofilaments, and then ultimately the microfilament here is you get the 4-1 ball weave together. Here's another way of, like, if we take a cross section of a hair. Here's our first alpha helix. There's our coiled coil, and we made the protofilament. These guys, then they give kind of slightly different names to the basically higher ordered structures in here. And ultimately, you have a hair cell in there. Now, then these higher order structures are held together by disulfide bonds up here. Okay, so here's the next question is, what do you do if you want to curl your hair, okay, or straighten it for that matter, because if your hair has a certain structure, basically, that structure is locked into place with a disulfide bond. So what you do is you treat it with a reducing agent to reduce those disulfide bonds, then either curl it or straighten it, depending on what you're trying to do, and then reoxidize those bonds, and then it'll basically lock it into its new shape. Here it's basically showing for curling hair rather than straightening it, straightening hair. So, of course, these reducing elements they're kind of smelly stuff, so that's why this process is a little bit smelly in the hair salon. So this is back in my generation. This is like Farrah Fawcett. That was the big thing, putting in the curls in there. Even Burt Reynolds. Anybody remember Burt Reynolds? Yeah, there's Burt Reynolds. Smokey and the Bandit. He got the perm going. That was a big thing. Then later on it was the other way around, right? Got to take the curls out, make it all straight in there. But it's not necessarily all just. All right, now we'll talk about collagen just a little bit, but I think I'll get into the details, but at least I'll introduce collagen here. All right. Collagen is the most abundant tissue in vertebrates, okay? And because it's in the connective tissue, it's basically in your, it's part of the, it's part of the ends of the bones and things that attached bones are. What collagen is if you make chicken soup, and you take the chicken and you boil it up for the day, right? And then you take it and you put it in the refrigerator overnight, and the fat comes to the top, right? And that's the creamy, and it solidifies. But if you take off the fat, a lot of times the cold soup is kind of jelly-like in there, okay? And it's that jelly-like stuff that's actually the collagen. That it's also, at that point, it's known as gelatin. It's basically collagen that you've extracted out of the connective tissue of the boils that you've been, of the bones you boiled up when you were making your soup. And then it basically solidifies once it gets cold. Now, if you're smart, what you do is you go through and you say, well, I can go to a slaughterhouse and I can get as much collagen as I want, and I can figure out a way to purify it out of there, and then make it nice and crystalline. I throw a little food coloring and sugar with it, and then you can make Jell-O. So that's basically what Jell-O is. It's gelatin, from extracted from various animals in there. The process was developed in like the late 1880s, and they've been selling it ever since. Okay, so what is collagen? Is firstly, it has an unusual helical structure, and it has an unusual helical structure because it has mostly three amino acids. Mostly glycine. What do we know about glycine? That's one of the most conformationally flexible amino acids. Proline. It's a secondary amino acid, which actually locks it into a particular structure, and a derivative of protein known as hydroxyprotein. Okay, let me just finish up by telling you the story. Hydroxyproline requires the oxidation of proline. It's a post-translational modification, okay, and it ends up the enzyme that does the hydroxylation requires ascorbic acid, vitamin C. So if you lack vitamin C, the disease that you get from the lack of vitamin C is known as scurvy. One of the first things is your collagen begins to break down. You do things like your gums break down, you start losing teeth and that sort of thing. scurvy was a problem on old sailing ships. You guys know, you guys heard of Captain Cook in the Endeavor at all?
That's the one with, like the tomatoes, that was in the can, you found out how much it was saved, it's great.
No, I don't know. No, he was an explorer in the late 1700s. The British explorers explored the South Sea. He went down there several times. He eventually got killed in Hawaii. But anyway, he's also the model. So Captain Cook, he had, he was an explorer going where no British men had been before. He was the model for Captain Kirk in Star Trek. Captain